Hello everyone, welcome back to my channel. Today we will discuss about the topic nucleic acid. It can store the genetic information that transmit the heredity from one generation to another generation. It can direct the protein synthesis and it is essential for life. It is the molecules of the heredity. By the way, myself Dr. Tithli Sadhu, Associate Professor of Department of Biochemistry, School of Agriculture, SR University, Warangal, Telangana. Let's start today's topic. Nucleic acid. It is the molecules of the heredity. It actually helps us to know that how does the chemistry of the life work. From this lecture, we can know that why do children resemble their parents? Where is the genetic information stored inside our body? Being a very small molecule, how can DNA can carry so much of information from one generation to the another generation? Why do cells need both the DNA and also the RNA? What are the DNA and RNA actually made of? And how can the nucleic acid chemistry be used in the medicine in the modern era for any sort of the gene related issue? Nucleic acids are the large biological macromolecules that can store, transmit and express the genetic information in living organisms. It's actually built up of long chain of polymers that calls the nucleotides. The tides that means three. So it is made up of three components, that nitrogenous base, pentose sugar and phosphate group. The detail about these three we will discuss in this lecture step by step. Nucleic acids were first discovered in 1869 by the Friedrich Meischer. Nucleic acid actually two types. One is DNA, that is the deoxyribose nucleic acid, and another one is the ribose nucleic acids RNA. The demonstration that DNA contained the genetic information was first described in 1944 by the Avery, MacLeod and McCarry. DNA actually acts as a permanent genetic material in our body, whereas the RNA performs the several roles for protein synthesis, gene regulation, and many others activity. So that's why the body needs both the DNA and also the RNA. Whereas DNA acts as a book and RNA acts as a worksheet for the regular work. Nucleic acids are present in all the living cells and many viruses also. The main cellular sources of the nucleic acids are the nucleus, mitochondria, chloroplast, cytoplasm, ribosomes and viruses also. Viruses can contain either DNA or RNA as their genetic material. The dietary source of the nucleic acids actually occurs in all the cells, but they are naturally present in the fish, meat and eggs, the milk and dairy products, pulses, beans and with different types of vegetables and fruits and mushrooms, yeast, etc. What are the importance of the nucleic acids? It is essential for the continuity, growth and functioning of the life, because it can transmit the genetic information from one generation to another generation. It is actually helping to know the chemistry of our life. So without the nucleic acid, nothing can be possible. They preserve the hereditary information from one generation to the next. They determine the structure and function of the cells. They provide the instruction for the synthesis of the protein. They control the growth, the development, reproduction and metabolism of all the body. They generate the genetic variation through the mutation and the recombination. They help the organism for adapt and evolve. They form the molecular basis of the biotechnology, genetic engineering, molecular diagnosis, forensic science, modern medicine, gene related issues and all. The analysis of the DNA and RNA helps to identify the genetic disease, and nowadays the gene changing type of health science are also going on. Then the central dogma, that is the very important things of our life. It is a part of the molecular biology. It is a theory that stating the genetic information flows not only in one direction. It flows from DNA to RNA, RNA to protein, and RNA to directly to protein, like that. The DNA itself can replicate to DNA, or through DNA it can be possible to produce the RNA through the transcription process, where the DNA used as a template to make the RNA, and the RNA again through the translation process is converted to the protein. So this is complete the process of the central dogma, that is the main part of the nucleic acids, and without the central dogma no life can be possible. Now we can come that the difference between the DNA and the RNA. The DNA stores the complete genetic information of an organism, whereas RNA helps us to use this genetic information that's stored in the DNA. That's why I told already, if DNA is a book, RNA is the worksheet. From that book we have to do. DNA acts as a long-term and stable repository of the genetic information, whereas the RNA serves as a temporary carrier of the genetic information. DNA replicates before cell division to produce the identical DNA copies that known as the replication. Generally, the RNA does not self-replicate except in some RNA virus. The DNA transfers hereditary information from one parent or one generation to the another generation, whereas RNA transfers the genetic information from the DNA to the sites of the protein synthesis. DNA provides the original genetic code for making the protein, whereas the RNA directly participates in the protein synthesis through the messenger RNA, transfer RNA, or the ribosomal RNA. The DNA does not normally carry the genetic message from the nucleus to the ribosome, whereas the mRNA carries the genetic message from DNA to ribosome. The DNA does not transport the amino acid. The transfer RNA transports the specific amino acids to the ribosome at the time of the protein synthesis. DNA does not form the principal structural component of the ribosome. rRNA, the ribosomal RNA, combines with the proteins to form the ribosomes. Usually does not act as a biological catalyst, the DNA, whereas some RNA molecules act as a catalyst that are called as ribozymes. The DNA contains a regulatory sequence that controls when and how genes are expressed. But the regulatory RNA such as the miRNA, siRNA control the gene expression and the protein production. The DNA indirectly controls the growth, metabolism, development, reproduction by directing the protein synthesis, whereas the RNA carries out and regulates that many instructions that can help for the growth and metabolism, development and all. The DNA help for the genetic variation. The mutation and recombination in the DNA produce the genetic variation. RNA helps for the gene expression. Genetic material in viruses, the DNA serves. RNA also serves the genetic material for some viruses, that is RNA virus. And overall, we can say that DNA is a genetic blueprint, whereas RNA is a working molecule. Actually, DNA and RNA both are very important for our body. We cannot exist with one only. The DNA acts as that permanent storage molecule of the genetic information, whereas RNA helps us to use those genetic information for the protein synthesis and gene regulation. Actually, the DNA and RNA, they act as a complementary. DNA is the information storage. RNA is the information utilization. DNA is a book. RNA is a working copy of that book. What are the components of the nucleic acid? We told already that nucleic acids are made up of the repeating unit of nucleotides. The nucleotides, that means the tides is equal to 3. So 3 components are there. What are the 3 components? That is the nitrogenous base, pentose sugar, and one or more phosphate group. We can say that it is the ribose sugar and it is the nitrogenous base, whereas it is the phosphate group. So these 3 components should be there. Then the total sum we can call that it is a nucleotide. If we remove the phosphate from the nucleotide, if we remove the phosphate from the nucleotide, then the nitrogenous part and the sugar is equal to nucleoside. The pentose sugar actually 2 types, that it can be deoxyribose and it can be ribose. The nitrogenous base, it is 2 types. It can be purines, it can be pyrimidine. And the phosphate, it can be 1, it can be 2, it can be 3. The nucleotides in nucleic acids are linked by the 3' 5' phosphodiester linkage. That nucleic acids are the polymer of nucleotide. So one nucleotide is bind with the another nucleotide by 3 to 5' phosphodiester linkage. How? This is one nucleotide, that it is the sugar carbon. Here the nitrogenous base, maybe it is cytosine. It is one phosphate group, whereas this one is the another nucleotide, that is it is the sugar molecule deoxyribose, that thymine is linked here and the phosphate group is here. The 3' carbon of this sugar molecule is attached with the 5' carbon of the another nucleotide molecule. So that's why this bond is known as the phosphodiester bond, and the direction is from the 3' to 5' direction. That's why it can called as a 3' 5' phosphodiester linkage. There are 5 bases present in the nucleic acids that have the carbon nitrogen ring, so that's why it is called the nitrogenous base. These are 2 types, one is purine and another one is the pyrimidine. The adenine and the guanine are called as the purine nitrogen base, whereas the thymine, cytosine and uracil are called as a pyrimidine nitrogenous base. The purine are the bicyclic compounds consisting of a pyrimidine ring fused with an imidazole ring. That it is the purine. In this purine, it is pyrimidine ring that is fused with an imidazole ring. How the numbering of the purine is happening? The purine ring system is numbered starting with the bottom nitrogen of the 6-membered ring as 1 and proceeding counterclockwise around the 6-membered ring to the position 6. So it is 1, 2, 3, 4, 5, 6. And moving to the adjacent carbon in the 5-membered ring as 7, 8, 9. Junction atoms are skipped. The numbering starts on the larger 6-membered ring and goes counterclockwise, then shifts to the smaller ring and goes clockwise. Whereas the pyrimidine at the monocyclic compound consisting of a 6-membered ring with the 2 nitrogen atoms. And here the numbering starts from the bottom nitrogen as a position 1 and then proceed clockwise around the ring so that the heteroatoms and the functional groups get the lowest numbering. Here the nitrogen position is the 1 and at the 3 position. Then what are the major bases in the nucleic acids? That purine and the pyrimidine are the main. Purine is a 2-ring base and the purine is 2 types. One is the adenine and another one is the guanine. The adenine in the 6th position, the amino group is attached. So it is known as a 6-aminopurine also. Whereas in the guanine, it is known as the 2-amino and 6-oxypurine. The pyrimidine is a 1-ring base. It has 3 components. The thymine, that is 2,4-dioxy 5-methylpyrimidine. Uracil, that is 2,4-dioxypyrimidine. And another one is the cytosine, that is 2-oxy-4-aminopyrimidine. What are the sugars of the nucleic acids? Nucleic acids contain the 5-carbon monosaccharides, which are known as the pentose. RNA contain the D-ribose, whereas the DNA contain the 2-deoxy-D-ribose. The sugar differs only at the 2' carbon. Actually, for the RNA, that is the D-ribose sugar, at the 2' and 3', both position the hydroxyl groups are present. But whereas in the case of the DNA, that is the 2-deoxy, so this 2 position OH is omitted. So only at the 3' position the OH is present. So that's why it is the deoxyribose and it is D-ribose. Nucleotide is a phosphate ester of the nucleoside. I already told if you remove the phosphate group from the nucleotide, you will get the nucleoside. So the nucleotide is a phosphate ester of the nucleoside. It consists of a phosphate group joined to a nucleoside at hydroxyl group attached to the C5 group of the sugar, that is 5' nucleotide. In DNA, it is known as the deoxyribonucleotide. For RNA, because of the presence of ribose sugar, it is known as the ribonucleotide. Deoxyribonucleotides and the ribonucleotides can have a single phosphate, 2 phosphate and 3 phosphate group. So we can say that without this phosphate group, if only the nitrogenous base and pentose sugar is present, then it is known as the nucleoside. If this nucleoside is attached with this phosphate group, then it is known as the nucleotide. If here only 1 phosphate group is present, then it is monophosphate. If 2 groups are present, it is diphosphate. And if 3 phosphate groups are present, it is the triphosphate. And the phosphate group is attached, actually the 5' position, means the C5 group of the sugar. That's why all the nucleotide, by default, most of the cases, it is the 5' nucleotide. Then what is the function of the nucleotide? It is a built-in blocks of the monomeric units in the nucleic acids, store and transfer the genetic information, involved in the energy transfer, participate in the cellular signaling also. So it is the structure of the nucleotide monophosphate, nucleotide diphosphate and nucleotide triphosphate. So all the cases it is the nucleotide, not deoxyribonucleotide. So the ribose sugar is present. So you can see at both the 2' position and the 3' position the hydroxyl groups are present. So all the bases it should be adenine, guanine, cytosine and uracil, because thymine is exclusively for the DNA. So if we come to the deoxyribonucleotide, so here we can see that 2 position the OH is missing because it is the 2-deoxy. So that's why in the 2' position the hydrogen is present, only one hydroxyl group is present among these 2' and 3', and the bases should be the adenine, guanine and cytosine and thymine, not the uracil. Uracil is exclusively for the RNA, whereas the thymine is exclusively for the DNA. And here in the monophosphate case, only one phosphate group is present. For the diphosphate, it is the 2 diphosphate groups are present, and whereas in the triphosphate, 3 phosphate groups are present for both the case of ribonucleotides and deoxyribonucleotides. Then the structure of the things, that is the adenine, that is one purine base. If it is added with ribose, it is the adenosine, one kind of nucleoside. That is, it is our ribose sugar. Always the one position, the C1 position is attached with the N9 position of the adenine. So it is actually happening that ribose sugar is attached with the N9 position of the adenine. It is known as the adenosine. And if the phosphate group is present, then it will come that is the adenosine monophosphate. So the C5 position is attached with the phosphate group. Similarly, for the adenosine diphosphate, the C5 group is attached with the 2-phosphate group, and the C1 position is attached with the N9 of the purine. And for the same in the adenosine triphosphate also, the C5 position is attached with the 3-phosphate group, and the C1 position is attached with the N9 position of the purine groups. The phosphate, if it is very close, then it is known as the alpha phosphate. This phosphate is known as the beta phosphate, and if 3 phosphates group are present, then this first one is alpha, this one is beta, and this one is known as the gamma phosphate. Then the structure of the guanosine triphosphate, it is the structure of the cytidine triphosphate. It is the uridine triphosphate, and it is the deoxythymidine triphosphate. So all the cases we can see only the bases are changing, all other structure part is same. This structure part is totally same, that is, and all are triphosphate. So everywhere 3 phosphate group are attached with the C5 position of the sugar molecule. So for the guanosine, cytidine and the uridine, we took the ribose sugar. So everywhere we can see that 2' and 3' position both are attached with the hydroxyl group. For the guanosine triphosphate, the guanosine, that purine group is attached with the C1 carbon, and for the cytidine, that is the cytosine, that pyrimidine group is attached with the C1 carbon, and for uridine also the same thing. And deoxythymidine, it is where we can use the deoxyribose. So here in that case 2' is not contain the hydroxyl group, only 3' is containing the hydroxyl group, rest of the part is same. Then the nomenclature of the nucleotides, we can say that adenine is the base and ribonucleosides is the adenosine. If the sugar is ribose, so it is ribonucleoside, and if it is 2-deoxyribose as a sugar, then we know it is the deoxyribonucleoside. If phosphate is attached with the nucleosides, it will come as a nucleotide. The single phosphate gives a mononucleotide, 2-dinucleotide, and that is diphosphate, and if it is 3, then the triphosphate. So we can say that if adenine has a base, so ribonucleotide is a adenosine, and for the deoxyribonucleoside, if it is act as a sugar, then we can say it is the deoxyadenosine monophosphate, deoxyguanosine monophosphate, like that. And these hydroxyl groups of the nucleosides are esterified with the phosphates to produce the 5' or 3' monophosphate. So actually these 5', these groups is actually most commonly esterified. So most of the cases it is the 5' usually, but whereas in the case of some cases, if the 3' is attached with the phosphate group, then it is known as the 3' adenosine monophosphate or guanosine monophosphate, like that. So based on the carbon number, the 3' of the or the 5' we can decide. After the nucleoside, that if only the nitrogen base and the sugar molecule is present, then we can say it is the nucleoside. In RNA, the nucleoside have the ribose as a sugar, so it is known as the ribonucleoside. But in the case of the DNA, it is present at the 2-deoxyribose molecule, so we can say it is the deoxyribonucleosides in the case of the DNA. And every time the C1 carbon of the ribose sugar or the 2-deoxyribose sugar is attached with the N1 position of the pyrimidine and N9 position of the purine base. So in the case of the adenine and the guanine, every time the N9 position is attached with the C1 carbon, whereas in the case of the thymine or cytosine or the uracil, the N1 position is attached with the C1 carbon. Then we can say the structure of the adenosine, that we can tell that it is the ribose sugar, and the C5 carbon or the C5 carbon of this ribose sugar is attached with the OH group, and later these only it can make the phosphoester bond where the phosphate groups can attach, and this C1 position is attached with the N9 position of this adenosine. Then similarly for the deoxyadenosine, only in the 2 position only the hydrogen is present, the hydroxyl groups is not present, and here also the C1 position is attached with the 9 positions of the adenosine. Then we can say that is the cytidine. For the cytidine also it is the pyrimidine base, so the N1 position is attached with the C1 group. Similarly for the guanosine, again the N9 position is attached with the C1 groups of the ribose sugar, and the same case for the uridine and the thymidine also. But you have to remember that for the uridine case, every time we should take the ribose sugar, because uracil is exclusively for the RNA, whereas the in case of thymine exclusively it is for the DNA, so we should take that is the 2-deoxyribose sugar. Then that the tautomeric forms of the purine and the pyrimidines, that tautomerism means the reversible movement of a proton with rearrangement of the double bonds. Actually, if the keto groups or the lactane form is present, so that there itself the tautomerism can possible, because that here is the possibility that this keto form can convert it to the enol form, because these will come here and it will actually go to the upward direction, so we can form the COH plus C double bond N. So this ketoenol and lactam to lactim conversation is the possible. The ketoenol tautomerism we can say for the guanine case, but in the case of the cytosine, that is the lactam to lactim, and in the thymine and uracil also, that keto to enol tautomerism case we can see. After that, chemically modified forms of the standard bases, because there are 5 standard nitrogen base. One is the adenine, guanine, cytosine, thymine and uracil. But apart from these also, some chemically modified standard bases are coming, either through the methylation or the acetylation, or by changing the isomers we can get the different types of the different minor bases. That's actually help for the gene regulation or some the gene modification and all, and also help sometimes for recognition of some specific enzymes and the proteins. So in the medicinal term is actually very much useful. This kind of some bases examples are the 5-methyl cytosine, that's found mainly in the DNA. Its major significance, the gene regulation and the recognition. That acetylosine is also one kind of modified base, is actually can present in RNA and the DNA both. Stability and the recognition is the major significance. Then methyl adenine is also possible. Pseudouracil is possible. It is actually exclusively for the RNA. Stability and the accurate recognition is possible from it. After that, we can say that purine bases are also present in the plants, that is the methylated purines, because normal purine skeleton is there. If we add some methyl groups, either 1 or 2 or 3, we can get the methylated purines, and those purines have some biologically active compound, like in the caffeine, then in the tea, that is the possibility of the theophylline, then in the Cadbury the theobromine, all are kind of the purine base, but the methylated purine base, so the modification is there, and those all are very very useful for the body also. And that purine pyrimidines and some nucleotide analogs are nowadays is very common in the modern medicine technology, because the synthetic analogs are made by modifying the rings or the sugar moiety. Actually it is possible to modify either the ring or the sugar molecules to get the nucleotide modification, and the modified nucleotide actually sometimes helpful to inhibit the enzymes or block the DNA RNA synthesis or stop the cell proliferation. So mainly for any type of genes case issues, genes health issues, or in the case of the cancer, many critical disease nowadays, that's why the medicines or some medicinal application is possible, because of this gene therapy is possible now because of this analogical change. So these are the different types of the analogs or drug examples are there, like the allopurinol, that the 5-fluorouracil, marcaptopurine, etc. That's actually help for the cancer therapy, or some the guts problem, or some actually diet adenosine is helpful for the AIDS or HIV therapy. So previously what are the disease are there, we cannot imagine that we can treat those disease. Nowadays for this type of analogical change of these nucleotides or nucleosides, it is very much possible nowadays. So that's the way that how the nucleotide is helpful for the modern medicinal chemistry also. Thank you. So in this from this topic, if you have any doubt, any clarity or something, please comments and do subscribe and like for this video. Thank you.